All right, so welcome back to part two of our histology lecture. So in this part, we're going to look at stratified squamous, and then um, we're going to look at a couple of example images of epithelial tissues. So we talked about in the first part, we talked about simple tissues. So simple squamous, cuboidal, columnar, and then we talked about pseudostratified, right? So now we're going to look at stratified tissues. We're really going to look mainly at stratified squamous, okay, because that's kind of the most common one that we run across. So when you look at this tissue, first off, just based on the name, stratified, we know that we're looking at more than one layer, okay? So at minimum two. For stratified squamous, usually we're looking at a lot more than two, okay? For stratified squamous, what we're going to have is the underlying cells, the very basal layer of cells. These are going to be alive, okay? And the cells at the top that are on the surface of the epithelial tissue, these are going to be dead, all right? Now, for stratified squamous, sometimes the cells can change shape as you move up, all right? So the cells at the bottom, at the basal layer, usually they're more cube-shaped, they're a little more cuboidal, and these are mitotically active, right? So this idea of being mitotically active means that they are rapidly dividing. They have a blood supply, they're being nourished, they're undergoing mitosis, okay? As they divide, they push their way up toward the apical surface of the epithelial tissue, so toward the kind of top, more superficial layer. And as they move up, they become more and more flat. So they become progressively flat. They lose their nucleus, they lose their organelles, and they ultimately die. Okay. So the cells at the top. So think of your skin. If you touch your hand with your opposite hand, what you're touching is the most superficial apical layer of that epithelial tissue, and those cells are dead. Okay, so when you touch your skin, those cells on the surface, those are dead skin cells. All right. If you move down in your hand toward the basal layer, and we'll talk about the specific layers of the epidermis in a later lecture when we do the skin, but those layers down at the bottom called the stratum basal of the skin, those are very much alive. They're very much alive. They're dividing, undergoing mitosis. Okay, so we said earlier that a simple squamous, very thin, allows for things to move across, gases and different things whenever we're filtering or whenever we're reabsorbing things maybe in the kidney or something, right? So when we have stratified now, kind of think the opposite, right? We have lots and lots of layers, so this very stratified, this very thick layer of epithelial tissue, and so that probably means that we're going to be more focused on protection and preventing damage to underlying tissue, okay? And that's exactly what we see here. So with the stratified squamous, we have two flavors, okay? We have two types. We have stratified squamous that is keratinized, and we have stratified squamous that is non-keratinized, okay? The keratin, this is a protein, okay, that's on the surface of the cell, and we'll talk about it more when we get to the skin, but it can serve as a kind of waterproofing, water-resistant type protein, and it serves to protect the overlying layer. So if you have stratified squamous that is keratinized, so keratinized stratified squamous epithelium, this is our skin. Okay, this is our epidermis, right? So when you look at your skin on your hand, that is a keratinized stratified squamous epithelium. Our non-keratinized stratified squamous epithelium is all the other epithelial tissues that are stratified squamous in the body. So think of the inside of your mouth, the inside of your cheek. When you ran your tongue on the inside of your cheek, that is a stratified squamous epithelium. It's non-keratinized. Okay. When you swallow food and it goes down your esophagus, that is a stratified squamous epithelial tissue that is also non-keratinized. All right. So if you look here at the kind of general appearance, you can see that you have cells at the bottom, at the basal layer, very kind of round and cube shaped, and then as you move up in the cell, or in the epithelial tissue, they become progressively flat, and they progressively lose their organelles and their nucleus. Okay, so here you can see what we just talked about. You find this in the skin, lining the esophagus. So this was keratinized, this was non-keratinized. You can also find it lining the vagina. In this case, it would be a non-keratinized. You can find it lining the anus as well. And then we have it found lining the inside of the mouth. Again, all of those are the non-keratinized stratified squamous. You'll see another term if you read in the literature or read in books or watch videos or something, the term cornified. Okay, cornified and keratinized, those are kind of analogous. Those are essentially meaning the same thing. Okay, so this image in the top right, although it's a schematic, it's not exactly 100% accurate. It gives a good demonstration of the cells at the bottom being roughly round and kind of cubed, and then as they move up, they get flat, they lose their organelles, and then you can see the ones on the top are dead and they're flaking off, right? This is a good example of what we see in the skin, okay? All right, so the next one, and this last one that we'll talk about before we look at some images, is called transitional epithelium, okay? There's another name for this, and it's called urothelium, all right? So urothelium is somewhat of an older name, an older term. But you can use transitional, you can use urothelium, either one, okay? So this idea of transitional epithelium, transitioning, kind of implies that maybe the cells are going to change shape and they're going to transition from one to the next or something like that. And that's kind of true, all right? So where you find this epithelial tissue is you find it lining areas of the urinary system, okay? Hence the term urothelium, okay? So specifically, and the most common place you think of it, is lining the inside of the bladder, okay? So this image down to the right, this white space in the middle, that's the lumen of the bladder. That's the inside of the bladder. And then the outside here, this is the epithelial lining of the bladder, okay? You also find it in the ureters. You find it in parts of the urethra as well. You'll talk about that when you get to the urinary system, okay? So these cells can change shape. Now think about it. Why? Think about it in context of a bladder. Why do we want this epithelial tissue to change shape? Why is that significant? Why is that important at all? Well, in structures like the bladder, you can have nothing in the bladder, right? So it can be void of urine. There's nothing there. It's collapsed. Or it can be very full of urine, right? So depending on the volume of urine in the bladder is going to need to stretch and change size in order to accommodate that increase in liquid, right? In this case, urine, right? So same thing in the ureter, same thing in the urethra. As you increase the amount of fluid there, increase the amount of urine, you're going to need to stretch the lining to allow it to accommodate that, right? So usually these cells can be relaxed, they're kind of dome-shaped, and then they can flatten out as we stretch and as we fill up the bladder with increased amounts of fluid, right? So here you can see two different images. The image on the top is when the bladder is empty. It's relaxed. There's not a bunch of fluid there. And then in the bottom, this image here is showing what the bladder looks like when it is full, when it has a significant amount of urine in it, right? So in the top, you see that they're roughly kind of round and maybe kind of cube-shaped, and they go all the way up. They're kind of tall. And then whenever you fill it with urine, it stretches it out. So it goes from being really tall and it kind of squishes it down, right? Imagine you had like a rectangular section of Play-Doh or something, and then you push your hand on it, right? That Play-Doh is going to now stretch out and flatten out, right? So now in the bottom image, when we flatten it out because we've added urine, right? We've added weight to it. We've added this liquid. It's going to stretch it out, and these top layers of cells are going to become a little bit more flattened, okay? So you find this in the ureter, bladder, urethra, and the big idea here is it's allowing it to stretch to accommodate this increase in fluid, okay? The important thing is also it can return to its normal shape, right? So you can stretch it out when it's full, and then when you urinate, you empty the bladder, and that bladder can kind of collapse back, okay? You'll look at other features of the bladder when you do urinary system, things called rugae on the inside of the bladder, these kind of folds in the epithelium that also allow the bladder to stretch and accommodate an increase in volume. Okay, so before we look at a couple of examples, we also have epithelial tissues making up glands. Okay, so we have different types of glands based on how they secrete products and where they secrete them. Okay. So you have what are called endocrine and you have exocrine, okay? Endocrine, by definition, they secrete their product into the blood, okay? In this case, it's going to be hormones, right? Exocrine, and kind of what we'll focus on in this context because we're talking about epithelium, exocrine secrete their products into a duct, so D-U-C-T, a duct, and then from that duct, the product goes onto the epithelial surface, okay? So they're lined with epithelium. This is an epithelial lined duct, and then they secrete their product ultimately onto an epithelial surface. So think of something like salivary glands, right? Salivary glands are composed of this gland, this glandular epithelium. They will secrete their product. So if you look down here in the middle, this is an example of a salivary gland. And then they secrete their saliva onto an epithelial surface. In this case, it would be the inside of the mouth, right? That inside lining of your cheek that we talked about earlier, that is an example of an epithelial tissue, and so that saliva can go there as well. Okay, you'll talk about exocrine, the exocrine pancreas when you get to the digestive system in AMP2. You'll also talk about the endocrine pancreas when you get there as well. Okay, so the way that these cells secrete their products can vary, and there's three types of secretion. There's merocrine, apocrine, and holocrine. Okay, and you can think about the way to differentiate these is what happens to the cell as it secretes it, okay, and the method of secretion. So in merocrine secretion, you have your product packaged into a vesicle, okay, so these little secretory vesicles that you see here, the product are these things in green. The vesicle fuses with the plasma membrane, right? So if you think of the chapter 2 lecture that we did, we talked a little bit about exocytosis. It fuses with the membrane and then it exocytoses the products out, okay? Nothing happens to the cell is not damaged or anything, okay? In apocrine secretion, you have your product packaged into, just the product, and then it fuses with the membrane, and then it is kind of pinched off from the plasma membrane, and it is surrounded by a portion of the plasma membrane. So you lose a little bit of the plasma membrane as the product is released because it is surrounded by the plasma membrane. Okay, so merocrine, this is an example of salivary secretion. This is an example of mammary glands secreting milk. And then lastly, the last one is holocrine secretion. So holocrine secretion is when you release your product, the cell itself ruptures, and the cell itself is essentially destroyed. So this is an example of secreting things like oil. Okay, so sebaceous glands. These are oil glands. When they secrete their oil, the top portion, the cell kind of fills up with the sebum, with the oil, and it ruptures. The cell bursts, and the products are then released into the duct, so into the oil gland duct, and then they go onto the epithelial surface, which in this case may be like a hair or something like that. Okay, so you have these different methods of secretion. All right, so let's look at a couple of different pictures and examples of epithelial tissues, and then we'll end this part and we'll move on to connective tissues. So for all of these, I'm going to show an image. You can pause it, try to identify it, and then I'm going to say the answer. All right, so as we get to them, just pause them and try to answer them before I say the answer. So this is an example of a simple columnar epithelial tissue, right? You know it's simple because you just see one layer. You know this is true simple versus pseudo-stratified because you see the nuclei in a single row, right? So you may find this in areas like the digestive tract or something like that. Okay, you may also find it lining the uterine tubes. In that case, they may have cilia on the surface. Okay, if this was in the digestive tract, you may have some microvilli. Okay, all right, so look at this one, pause it. This is an example of simple cuboidal, right? You see one layer of cells, and these cells are roughly cube-shaped. Okay, here, pause the video. This is an example of pseudostratified columnar, right? You see cell nuclei at varying heights. You can also see the presence of cilia on the surface. So this would be an example of ciliated pseudostratified columnar epithelial tissue. Okay, this here is an example of simple squamous, right? So we know this is simple squamous. We're looking at the alveoli. You see one cell on this side. You may see another on this side. So this is a simple squamous tissue. You find it in the alveoli. You may find it in the glomerulus and capillaries and things like that. Remember, very thin, allowing for diffusion of gases and things across the membrane. All right, to answer this one, this is an example of stratified squamous. Okay, now this is specifically non-keratinized stratified squamous because you do not see the presence of keratin on the apical surface. So you see kind of round cube-shaped cells at the bottom. As they move up, they become progressively more flat. They lose their nuclei. For the most part, some of them may still have it. So the ones at the top are essentially dead. The ones at the bottom are more or less alive. So you may find this lining the esophagus. You may find this lining the inside of the mouth as well. Okay. All right. Look at this one here. This is an example of transitional epithelium. Okay, now this one can be kind of tricky. So looking at urothelium, transitional epithelium, you have to look at a bunch of different images. But you see these large cells on top, and then you see these cells at the bottom that are more or less kind of flattened and kind of squished down. Okay, so this may be an example of a bladder as it's full of urine with those top cells kind of flattened. If you want to know, these top cells are sometimes called umbrella cells. They, like an umbrella, they protect the underlying cells from the acidic nature of the urine in the bladder, and then these cells underneath become kind of more and more compressed and flattened as the bladder fills. Okay, look at this example here. So this is an example of stratified squamous, but it is keratinized. So this is keratinized stratified squamous. So here you see the strands of keratin, this keratin protein, and then down below you see layers of the stratified squamous in this case. So this is what we would expect to find in our skin. Okay, in areas of thin skin. So we'll talk about the difference between thin skin and thick skin. But this is keratinized stratified squamous. So this is something you may find. You may find this on the arm. You could find this type of epithelial tissue on the leg, on the back, on the trunk, things like that. Okay. All right. So we're going to stop here with part 2, and then we'll pick up with part 3 by talking about connective tissues.